Okay, hi everyone. So today we're going to move on to chapter 7, and we're going to talk about cellular respiration. So in the previous class, we kind of talked about how producers can make their own energy source. And so today we're going to talk about us as consumer, what we do after we consume those producers, or other consumers that had consumed the producer previously, right? Such as when we eat beef, beef eats plants, so on. Okay, so we're going to talk about cellular respiration. So let's start off by just thinking about what is cellular respiration, right? So we tend to think of respiration as that breathing in and out. So it's like, why do we have to breathe in and out? Like, what is the purpose of that? Like, what are we hoping to produce with that? And what we're hoping to produce with that is pretty much ATP molecules for energy. So our body doesn't per se use carbohydrates for energy in that it doesn't use it directly, but it is going to use it by converting the carbohydrates that we eat into a source that our body knows how to use, or our cell knows how to use, which is ATP. And so in order to do this, we require glucose, which is the carbohydrate source, and we also require oxygen, right? So we have to actually breathe oxygen, and we'll see a little bit later what that oxygen is being used for. And so when we breathe in oxygen and we eat our food source, we're then able to produce a byproduct waste of carbon dioxide and water as we're converting that glucose into an energy source. So this is the reason why breathing is so important, is so that we can actually make ATP energy from the food that we eat. So we're basically reversing what the plants did. So the plants, they're taking carbon dioxide and they're taking water in order to make glucose. We are taking the glucose along with oxygen, because remember in photosynthesis, oxygen is a byproduct. Here, we're actually using oxygen. We're using glucose, and we're actually going to convert that to carbon dioxide and water as well as to energy. So let's think about this. What is respiration, right? So we kind of talk about how respiration is that breathing in and out. And what do we breathe in? Well, we breathe in oxygen, right? And we're going to breathe out carbon dioxide. Plants uses that carbon dioxide and it kind of quote unquote breathes, but it doesn't actually breathe, but it's going to break down the water and release oxygen as a waste product. And so our cells, inside cells, is doing the same thing. It's taking oxygen, making a glucose into energy, and then breathing it out. That's kind of, or it's releasing it. It doesn't actually, your cell doesn't breathe per se, but it's releasing that carbon dioxide. And it goes into your bloodstream and eventually into your respiration stream, and eventually you're going to breathe that out. So cellular respiration is the reason why we breathe. Okay, so what's happening here in cellular respiration is something called an oxidation and reduction reaction. And so what I want you to take away from this is that oxidation is when we remove hydrogen atoms and we lose an electron. Reduction is when we add hydrogen atoms and we gain an electron. So let's take a look at this equation out here. So glucose is that monomer, right, of saccharide. So this is our sugar. So glucose is our sugar. We're going to add oxygen, which is what we're breathing in. And what's going to happen is we have an oxidation reaction where we're going to remove hydrogen. So you can see that this hydrogen right here, C6H12O6, we're going to remove that hydrogen and end up with carbon dioxide. So we're removing hydrogen. In that process of removing hydrogen, we lost some electrons. And then our other reaction, because these reactions are always together, we're going to have a reduction reaction. What are typically together, but right here is together, right? So we have oxidation of taking the glucose and breaking it down into CO2 by removing the hydrogen. And then we have reduction, which is we're taking our oxygen source here. And what we're going to do is we're going to add hydrogen, right? So we're adding hydrogen to the oxygen. And in that sense, we're gaining electrons right here. And our end goal with this cellular respiration equation that I'm showing right here is to get that energy source. So this is how we go about breaking glucose down. So there's 4 phases of cellular respiration. The first one's called glycolysis. That's when we take our glucose and we convert it to this other molecule called pyruvate. Our preparatory reaction. So in order for us to be able to actually take that pyruvate and use it downstream to make energy, we have to kind of prepare it into another form. So we're going to take our pyruvate and we're going to do a preparatory reaction where we convert it to this other molecule called acetyl A. And then that's what we're going to actually pass onto the citric acid cycle. So the citric acid cycle is the cycle where we're going to take that molecule that we made. We're going to go through it. In that process, we're going to make some ATP. We're going to make some NADH as well as FADH too. And then from that, we're going to move on to our electron transport chain. And electron transport chain is the last part of our cellular respiration. And that's when we're actually going to really make ATP. So what I want to take away from this is there's four phases: glycolysis, preparatory, citric acid, and the electron transport chain. Notice that along the whole process, we're moving electrons along the way. So think of this electron as that energy source. So we're breaking this into smaller reactions so we can capture the most energy, because glucose, if we were to break it directly, we would lose a lot of that energy as heat. So think about that marshmallow hitting that fire, and there's so much energy in that marshmallow. But if we try to break it down right away, like when you light it up for s'mores, you see that the marshmallow just lit right on fire because there's just so much energy. So we're trying to avoid that happening by breaking into smaller steps. So we're going to break it into these smaller steps. And in that step, the eventual step is the electron transfer chain. That's where we're going to make the most energy, convert most of that electron energy into ATP. But along the way, we do mix a little bit. So like glycolysis, we make a little ATP, we make two ATP molecule. Through the citric acid cycle, we're gonna make some ATP, so we make two ATP molecules there too. But then when we get to electron transport chain, look at that number, 34. We are able to make 34 ATP at this point. So let's first look at like glycolysis. So glycolysis is going to happen in the cytoplasm. That is, it's going to happen outside of the mitochondria organelle. So it's going to happen in cytoplasm. This is true for both prokaryote and eukaryote. And we're basically going to take the food source that we ate. So think of, when you eat like a loaf of bread. We're going to take a loaf of bread. We're going to break that down to glucose molecules, so into monosaccharide. Then we're going to take that glucose molecule and go through glycolysis. Glycolysis takes that glucose molecule, it breaks it into, so glucose is 6 carbon. We're going to break that into 2 3-carbon molecule. This is called pyruvate. So we're going to break the glucose into pyruvate. So it goes from a 6-carbon into 2 3-carbon. And it takes some energy to be able to do this, but we're also going to harness some energy. So it takes some energy for us to be able to convert that glucose into this pyruvate molecule, but at the same time, the energy of breaking those bonds, when we're going from that 6-carbon to the 3-carbon, we're actually going to also harness that energy, and we're going to harness it and place it into ATP. And this is, again, happening outside of the mitochondria. So you don't have to memorize all these big words. But what I would like you guys to recognize is that we start with glucose up here, and glucose 6-carbon, and we're going to break into 2 3-carbon. So glyceraldehyde 3-phosphate is what G3P is. So we're going to take the glucose and we're going to actually break into 2 of these glyceraldehyde 3-phosphate. This takes quite a bit of energy for us to actually do. So you can see here that we're taking energy right here in order to convert this glucose into another different molecule. So it's just changing the different molecules. We can see that change because it adds these phosphates to it from the ATP. And then it's going to break into 2 smaller molecules. So now these two smaller molecules ready for us. And this is the whole reaction here. What I want you to take away from this and know is recognize that this takes some ATP to use up here, but we're able to get that ATP back in these reactions. So there's two G3P molecule, which means that we're going to do this twice. So here we end up gaining two ATP here and then another two ATP here, but we lost two ATP up here. So we're going to get a net worth of two ATP. And you can see that we're doing all these little reactions where we're changing the molecules basically in the end to get to pyruvate. That's our goal. All right, so this is a substrate level ATP synthesis. And this is going to produce 4 ATP, but because we use 2 ATP above, we have a net gain of 2 ATP. And we also make 2 NADH. And these are also known as electron carrier, the NADH. Okay, so what do we have? Let's review this again. What do we have at the end? So we input our carbohydrate source, which is our glucose. We have put in 2 NAD plus molecule that are going to be reduced later. And we had added 2 ATP, right? We also ended up with, so we're going to add 2 ATP. That's what I'm going to stop at that. What we actually end up with is converting the glucose to pyruvate. So we're going to convert to 2 pyruvate because the pyruvate is 3 carbon long. We're going to reduce our NAD plus by adding an electron to it and a hydrogen. And we end up with NADH. And then we're going to end up using some ATP, but we're also going to make ATP. So we're going to end up with a net worth of 2 ATP. So from here, pyruvate can go down a anaerobic process or an aerobic process. So which reaction it decides to go, which pathway is dependent on whether or not oxygen is available. If we have oxygen, the pyruvate is going to go into the mitochondria, and we're going to go down that citric acid cycle and that electron transfer chain route. If it doesn't have oxygen, pyruvate is going to go down a different pathway, which is called our anaerobic pathways. And we'll talk about that in a little bit. So here is a video. Let's just watch it real quick together.
Glycolysis, which begins the breakdown of glucose, is a series of 10 enzyme-catalyzed chemical reactions that can be divided into 2 main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into 2 3-carbon molecules of glyceraldehyde phosphate. The 3-carbon glyceraldehyde phosphate molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups. The phosphates are transferred to ADP, finally making some ATP. This way of making ATP is called substrate-level phosphorylation. A couple more reactions rearrange the atoms in the 3-carbon molecules. More ATP is generated in the final reaction that yields pyruvate. For each glucose molecule broken down during glycolysis, a net of 2 ATPs are formed, along with 2 NADH molecules.
Move on to the next slide. So that kind of tells you how we're converting our glucose to pyruvate. So we can go, if we don't have oxygen available, we go down this other pathway, which is to take our pyruvate and either go down the lactic acid pathway, which forms 2-lactate, or we can go down the fermentation pathway, which actually is similar to when you get alcohol fermentation, right? So in order to convert sugar, so let's say with beer, right? You need to convert that sugar into an alcohol. It has to not have oxygen available, and is basically taking that sugar and making it into energy source. And in that process, the byproduct is the alcohol. With the lactic acid pathway, it's really similar, where we're taking our pyruvate, and because we don't have oxygen, but our body still needs some kind of energy source, is going to undergo this anaerobic pathway in order to produce these two lactates. So the lactates, we're going to talk a little bit when we in the next step, but our goal is to take that electron. Remember how we're breaking the glucose up, and it's going to release all this energy in the source of these electrons. And what we want to do is harness that energy that's in those electrons. So we want to eventually pass on that electron to an electron carrier. So here we have lactate being in the electron carrier that we want to pass it to. And in this process, we actually will make some ATP. So we make a small amount, right? We're making 2 ATP. And you'll see later on that with the electron transport chain, we can make a lot more than just 2 ATP. So we're going to make 2 ATP, but we're also going to have this buildup of like the lactic acid. So if you've ever gone running, like see this woman right here, she's going running and you get that like pain that's kind of like on maybe around your ribs and stuff like that. It's probably your muscles trying to make some kind of energy source because it finds that it needs some sort of energy source, but because you're breathing heavily and you're not able to get oxygen too fast enough, it has that buildup, and that's kind of what that feeling that we have that you might sense it's a little bit pain. But once you start breathing again, you'll notice that that kind of dissipate because you no longer are unable to get oxygen to your mitochondria. All right, so let's kind of look a little bit more into these two steps. So we're looking at fermentation, which is what bacteria you will use in order to like I said, make energy without oxygen input. So we end up inputting glucose like before. The glucose is going to be converted to pyruvate like before. But now, because there's no oxygen, it's going to go down this pathway of the fermentation. And in that process, we're going to end up with the byproduct or the waste product of the alcohol, which was our electron acceptor and our carbon dioxide from breaking down the glucose. But we're able to produce some ATP as well. So we're able to produce 2 ATP. Because remember, we had used 2 ATP before and previously, but then we make more ATP in the process of the fermentation. So we actually end up with a net gain. And you see this with yeast as well, right? When we make bread and make it rise, and when we convert to beer, this is all a way for you to use the bacteria to break down sugar to make energy source, but they're just doing it without oxygen being the electron carrier at the end. Okay, so let's look at, we're going back to aerobic respiration now. So we're going to go back to this potato. It's actually a mitochondria, but it looks like a potato, doesn't it? So we're going to go to this mitochondria, and the rest of the phases is going to take place in mitochondria, and you need to have oxygen. So this doesn't take place if you're not breathing, if you don't have oxygen available. So glycolysis happens outside the mitochondria in the cytoplasm. Now we're going to go move into the mitochondria. And first we're going to go into the mitochondria matrix, and we're going to do the preparatory reaction, which is right here. The preparatory reaction is basically converting pyruvate into this acetyl A compound. So this acetyl A compound, what I want you to recognize from here is you're going to end up having 2 of these compounds because we have 2 pyruvate, right? Because glucose was 6 carbon, we're going to convert to 3 carbon. That this happens in the mitochondria, but in the matrix. And what's going to happen is the pyruvate is going to be oxidized. We're going to release carbon dioxide as a waste product. We're going to reduce NAD plus into NADH. This is going to be a way for us to carry the electron later into the next cycle. And we're going to take a 2-carbon acetyl group and attach it to the CoA to form acetyl-CoA. So this preparatory reaction results in us making our carbon dioxide waste product here, as well as 2 NADH and 2-acetyl-CoA that we can then use into the next reaction, which is the citric acid cycle. So let's go through first, and then I'm going to tell you what I want you to focus on. So this right here is going to also be in the matrix of the mitochondria. The acetyl-CoA is going to transfer its acylic group to this 4-carbon right here. It's going to transfer to this 4-carbon molecule. And this 4-carbon molecule is going to, because it has the acetyl-CoA now, produce a citric acid, which is a 6-carbon. The acylic group is going to oxidize to carbon dioxide. So you see right here, 1 molecule of carbon dioxide, and again right here, 2nd molecule of carbon dioxide. We're going to transfer some of the energy of the electrons into NAD plus and FADH to make NADH and FADH2 respectively. And then once we go through this whole cycle, the CoA is going to return to the preparatory reaction to be reused again. So it's a cycle because it kind of goes through the whole loop and it comes right back. Whoops, sorry. Oh my gosh. No. Ah, my goodness. Sorry, guys. Okay. So this is also a substrate level ATP, and we end up producing 2 ATP because we produce an ATP here, but since we're going to go through this cycle twice for every glucose molecule, you end up with 2 ATP. Okay, so what do I mainly want you to focus on? I want you to recognize that we start off with this 2-carbon, right? From glucose to pyruvate to acetyl-CoA. We end up with this 2-carbon molecule. We enter a cycle. We attach our 2-carbon to this 4-carbon molecule. So now we end up with 6-carbon. As we're breaking the 6-carbon back down in the cycle to a 4-carbon molecule, we end up releasing carbon dioxide. So we oxidize to carbon dioxide. And that gets released as basically a waste product because we're going to breathe it out. And we're going to make 2 ATP, 1 ATP per turn of the cycle. Right? So this is the point of the cycle. And the main reason why we go through the cycle is not really to make ATP so much as to get these NADH and FADH2 molecules that we're going to use in the next step for the electron transfer chain. So revisiting, oh, that was my kid, freaking rude. Anyway, sorry guys. The input is 2-acetyl-CoA, 6 NAD plus 2 FAD, right? So these are our electron carrier that we're going to add electrons to, and our ADP, right? So our output is going to be the carbon dioxide, which you saw was made during that cycle, right? One at a time, but we go through it twice. So we end up with 4 carbon dioxide. We're going to end up with reducing 2 NAD plus, not 2, sorry, 6 NAD plus, and we end up with 6 NADH because of that, and then 2 FAD, which will end up with 2 FADH2. And then we also make some ATP. So let's go through the cycle real quick so you guys can kind of watch the molecule goes through the cycle.
In the citric acid cycle, a 2-carbon compound reacts with a 4-carbon compound to produce the 6-carbon compound citric acid. In subsequent reactions, 2 carbons are lost to CO2, electrons are stored, and ATP is produced. By the end of the cycle, a 4-carbon compound remains, ready to react with another 2-carbon molecule and begin the cycle again.
I feel like that kind of goes through it pretty well, just so you can see that it's kind of reiterated. Although I will have to say this video makes me think that we're watching a horror movie at the same time. Okay, let's move on to the next part. No, I don't want to listen. So let's just kind of stop, take a little break right here and kind of see. I'll give you guys a few seconds to kind of see if you can remember what is happening at each stages, and then we'll go through it. Okay, so as we're going through each stage, let's just think about how many what our carbons are, right? So we're starting with glucose. Glucose is 6-carbon sugar. We're going to take that 6-carbon sugar and convert to pyruvate. At this point, pyruvate is still um, a 6-carbon molecule. We're going to take that 6-carbon molecule and break it. Sorry. It's a 3-carbon molecule. 3-carbon, right? We're going to take the glucose and we're going to break it down into 2 3-carbon molecules. So I meant to say that we still have 6 carbon total. But 6 carbon is going to go to 2 3-carbon. So now we have the pyruvate, which is a 3-carbon molecule. The 3-carbon molecule is going to undergo a few things, right? We're going to be making ATP in this sense, but it's going to undergo some reaction, and we end up with a 2-carbon acetyl-CoA. The acetyl-CoA is going to be added, that 2-carbon, to a 4-carbon molecule in a citric acid cycle. As we're going through, we're oxidizing, we're breaking down that original molecule 2 times. So that 6-carbon is going to be broken down. As it goes through the stage, it's going to be broken down so that we release 1 carbon dioxide, release another carbon dioxide. So we release 1, we end up with a 5-carbon sugar, release another carbon dioxide, and then with a 4-carbon molecule that would then be recycled. So we can do it again to an additional acetyl-CoA. So we end up doing this twice, but we go from 6 carbon to 3 carbon to 2 carbon to 1 carbon as we're breaking down our sugar molecules. How many net ATP are made at each stage? So if you remember glycolysis, we end up making just 2 ATP. The preparatory reaction, we use some ATP, we use 2, but we end up making 4. So we end up with a net gain of 2 ATP per glucose. That's what we're looking at, per glucose. And in the citric acid cycle, we will actually also end up, because we go through the cycle twice, we'll also end up with some ATP there too. Most of the ATP, we end up with 2 ATP. So most of the ATP is going to be an electron transport chain itself. We're also making some coenzyme. We're making NADH and FADH2 at each stage in the glycolysis preparatory and the citric acid. As I mentioned before, we are making these coenzymes because we have a lot of energy to store in the form of electrons, and we want to be able to place it in what I call these electron carrier, which is what NADH and FADH2 are. So we're going to want to move our electrons. So let's kind of go back so we can see the stages. I feel like this is always visually easier. Here we go. Right. So, oh no, we don't. Sorry, I'm trying to. I thought I had a plot that kind of shows us. Okay, I think this is where it's going, right? So we want to take all of these into these NADH. This is where we make the most NADH, is right here in the citric acid cycle. We go through this twice, but we end up with 3 right here and 1 right here. So 3 NADH and 1 FADH, but since we go through the cycle twice, we end up with 6 NADH and 2 FADH in the citric acid cycle. In our glycolysis cycle, where is it? I'm trying to find a picture because I find that I always have an easier time. So in our glycolysis cycle, we're also going to make and NAD, right? So right here we go from NAD plus into NADH. Since we have two pyruvate molecules, we're going to do this twice, which is why we end up with two NADH. We're not making any FADH, it's only NADH. So the only time that we make FADH2 is in the citric acid cycle. Okay, let's go back to here. So now this is the big finale right here is this electron transport chain. So this electron transport chain is going to take our coenzyme, which is our NADH and our FADH2. And remember, I said they're our electron carrier. So now they're going to deliver the electron to your electron transport chain. And as they deliver the electron, they're also going to deliver the proton, the H plus. So we're going to oxidize them so that we end up with NADH being broken down to NAD plus and F and H plus. So we're going to end with the electron. We're going to end with this hydrogen. This hydrogen right here is going to happen. This whole electron transport chain actually happens in the in the membrane, even though we can't see it here, because these two happen in the matrix, but then the electron transport chain is actually in the membrane. And what's going to happen is the electron from these electron carrier is going to be passed on to these protein carrier that's in the membrane. And each time we pass an electron, we're going to release some energy. We're also going to also add H plus into our intermembrane. And the whole point of this is to kind of have like a concentration gradient. So if you kind of remember when we were looking at situation in which we're looking at salt versus water and how water moves in and out of the cell, whether it's hypertonic or hypotonic. I just want you to appreciate that, there is gradient kind of happens, right? And in general, we tend to want to go with our gradient. So by adding H plus into the membrane and kind of pumping it in there, we're kind of causing there to be too much hydrogen. So what's going to happen is the hydrogen is going to want to leave and go with its concentration gradient. And it's going to do that at the last carrier. So when it does that at the last carrier, the electron that we've been passing along from glucose all the way down to this step, it's going to be finally added to the final electron acceptor, which is our oxygen. And this is going to form water. But in that process, all this H plus is also going to go through this molecule called an ATP synthase, which is an enzyme, and it's going to synthesize ATP. So the name kind of suggests, right? ATP synthase is going to synthesize ATP. So this is kind of what's happening here. What I want you to see here, like imagine this electron carrier, you're adding all this, we were adding the NADH and FADH2, which is our electron carrier. So it's carrying the electron right now. Electron is going to be passed along, and as it's passing along, we can end up making some we're going to be able to make some ATP, so 4 ATP in that first part, because you're going to have 2 NADH, 1 NADH per acetyl-CoA that was processed, or pyruvate that was processed through. We do that twice, which is why it's 2 NADH at this step. It's going to be able to make 4 ATP. We're going to do that again. And we're going to do that with each one. So you can kind of see that we're going to take the electron carrier, right? Because we have quite a few electron carrier at this point. We have 8 different electrons, 8. Well, we're going to have, hold on, 6 plus 2 plus 2. I can't do my math. 10, sorry. 10 NADH right here. That's going to be converted to ATP as it's passing its electron through. And then FADH is also going to come through. And the FADH isn't as rich of a molecule as NADH in that it doesn't carry as much energy for the electron. So you can see that NADH can sometimes make up to 6 ATP. In an ideal situation, it you end up with 38 oh no, 38 ATP. But realistically, in the cell, it tends to be less. It tends to be more like 34, sometimes 32, right? Sometimes 36. So it's more of that nuance has to do with more of the efficiency of which cell type it is. What I want you to recognize from this whole thing though is the role of NADH and FADH is to be the electron carrier and carry that energy over. And that energy that is carrying is going to be used to make ATP. And the majority of ATP is made in electron transport chain. And then we can make well over 30 30 ATP by transferring this electron down the electron transfer chain. And the transfer of the electron down the electron transfer chain also helps build this proton gradient, which is going to be, the proton gradient is going to be used to harvest the energy from hydrogen moving with this concentration gradient down to ATP synthase. So it's moving with this concentration gradient down to ATP synthase, and the ATP synthase will spin at that time and make some ATP. So as I mentioned, where that gradient comes from us forming this hydrogen plus concentration gradient. So when the two electron carrier NADH and FADH2 is donating its electron and its hydrogen to the electron transfer chain, the protons are just kind of being pushed against the concentration gradient, which is why it's an active transport because it doesn't want to move this way. It's going against its concentration gradient, which is what's forming that concentration gradient. So we're forming the situation in which we're putting hydrogen where it doesn't want to go. And the whole reason why we do that is so that the concentration now, because there's that concentration difference, it wants to go down its concentration gradient, and it's going to go down its concentration gradient within the ATP synthase interior right here. And as it's going down its concentration gradient, this ATP synthase is it's like a rotor. It's just spinning really fast. So it's taking that energy that's being the hydrogen going down its concentration gradient to like move this rotor, which will then take that energy source and transfer it to an ATP. So this is how we make ATP in our cell. And this is how the ATP is going to be used later to actually be used for all the other stuff that we need ATP for, right? Muscle contraction, standing, digestive, breaking up, or not breaking up, synthesizing like different bonds, right? That ATP is used a lot in our cell for a bunch of different things. But where do we get that ATP from? It's from this whole process right here that I've been showing you. Okay, so organization of the crystal. The crystal is, you can kind of see that this picture, right? The matrix is like kind of the inside. Citrate is this inner folding membrane right here. That's where I told you that the electron transfer chain is being located, because I told you that is there's this membrane right here. So this inner membrane has these proteins. These are the carrier proteins that I was talking about. So there's all these different little carrier proteins that's going to carry the electrons, or it's going to accept the electrons from the NADH and FADH2. So it's going to accept the electrons that these carriers are carrying. In the process of accepting the electrons, it's also going to push the hydrogen gradient against its concentration gradient. So this is active transport that is happening right here. This active transport will eventually lead of hydrogen being pushed will eventually lead to its concentration difference, which is what we see here. Bunch of hydrogen out here, very little hydrogen down here. So this goes down its concentration gradient. As it's going down its concentration gradient, you have this motor spinning, and that motor is going to result in ATP forming. So this is why we eat. This is why we breathe. So we eat so that we can get glucose molecule. We breathe so that we can convert that glucose molecule into actual ATP. We can make a maximum of 38 ATP, assuming that we're a standard, perfect, ideal situation, but some cells can make only 36 or even less, maybe 34, depending on the cell. Um, and this ATP that we make from glucose accounts for almost half of the energy that we are able to get from a glucose molecule. So glucose molecule, where do you guys think the rest of that energy goes? So let me ask you first. If it's able to harness 40% of the energy into making into ATP, where does the rest of the energy go? Oh, it's in the next line, right? A lot of that energy is lost as heat. So we would actually lose even more energy as heat if we didn't break it down to these smaller steps. Just because there's so much high energy that it could be almost like a combustion if we try to do it all at once. So we don't do it all at once. We actually take the glucose molecule, break it into all these smaller molecules so we can try to harvest as much as we can. And it's found that that's about 30 to 40% of the amount of energy that glucose is carrying. We're able to harness it and place it into ATP so we can do other reactions with it. And so are we able to get energy source from anything else that we eat? Actually, yes, we can. So we actually can get it from different fatty acid because we do have enzymes that allow us to convert it. We can actually convert uh, our our protein as well. So we can actually get energy from also fat and protein and convert it down this pathway to make ATP. It's just that the majority, because it's a little bit easier for the cell. The majority of your energy source, though, is going to actually come from the carbohydrate. And that's all I have for you for cellular respiration. So, main takeaway, actually, let me look at our study guide, make sure I cover everything. Sorry, let me see. Mm-hmm. Give me a second, you guys. Let's see, this is chapter 7. Okay, so we talked about the overall process, right? The overall process is to take glucose and convert it into ATP. What are the two main reactants and two main products? So I want you to think about both ways, both through cellular respiration as well as photosynthesis, what we start off with and what we ended up with. Describe the process of oxidation reduction. So think of that reaction that we talked about, where we're taking the glucose molecule and how we're going to break into carbon dioxide and water. So which one is the oxidation reaction, which one's the reduction reaction, and what's happening. The four main phases. So think of that one slide that we looked at, and we talked about what happens in the matrix versus what's going to happen, not the matrix, what's going to happen in the cytoplasm versus what happens in the mitochondria. And what are the four phases? So explain the energy investment, energy harvesting. So think about when we actually use ATP versus when we're actually able to harvest ATP. What happens to pyruvate depending on availability of oxygen? So remember that we're going to go from glucose to pyruvate. This is going to happen regardless if we go down the anaerobic route or the aerobic route. But once we get to pyruvate, whether we go down one route versus the other depends if oxygen is available, right? So what's happening? If oxygen is available, which pathway does it take? If oxygen is not available, which pathway does it take? Describe the role of the mitochondria. So what's happening in mitochondria with regards to the phases of glucose being broken down. So just kind of revisit that. What is the purpose of the citric acid cycle? So where does it happen to begin with, right? So at this point, we're in the mitochondria, but where within the mitochondria does it happen? And what is our main goal? So the main goal of the citric acid cycle isn't to make ATP. It has another kind of main goal that's related though to making ATP. So what is that? Explain the role of the electron transport chain, ATP synthase. We already talked about what the goal is, right? Once we exit the citric acid cycle, we end up with all this NADH and FADH2, and we still have our molecule as well. But the NADH and the FADH2, what is happening with those two once we actually hit the electron transfer chain, and how does it relate to um, production of ATP? And then how much ATP is typically produced from a complete breakdown of one glucose molecule? So think about if everything went perfect, how many molecules of glucose, not glucose, of ATP do we synthesize? If everything is not perfect, what's a more realistic number of how much ATP we can actually get? Okay, remember to do your quizzes, and if you guys have any questions, add it to the discussion board. All right, thank you, everyone. I will talk to you all soon. Bye.